Medical students, welcome to today's lecture on cell biology, where we will dive into the intricacies of the molecular mechanisms underlying neuronal birth and development, the navigation of the brain, pathfinding, building connections, wired functionality, neuronal polarization, chemical messengers, electrical excitability, sculpting connectivity, perfection in wiring, the importance of myelin, and decoding the brain. This comprehensive script aims to provide you with a detailed understanding of these topics, building a solid foundation for your future medical practice. Let's begin. In the early stages of development, neural stem cells divide to produce different types of cells. Factors like transcription factors, cell adhesion molecules, and signaling pathways play a role in this process. The progenitor cells eventually mature into neurons and form connections with other cells. Once neurons are generated, they need to navigate through complex cellular environments to reach their final destinations. This intricate process is guided by molecular cues, such as chemotaxis and contact-mediated repulsion. Growth cones, specialized structures at the leading edge of developing axons, respond to these cues by extending and retracting filopodia and lamellipodia, leading to axon elongation and path selection. Molecular guidance cues, such as netrins and semaphorins, provide attractive and repulsive signals, respectively, guiding axons along specific pathways in the brain. During pathfinding, axons encounter a multitude of cellular and molecular cues, influencing their navigation. These cues can be both attractive and repulsive, shaping the trajectory and connectivity of developing axons. Numerous molecules, including ephrins, slits, habosh, and their receptors, help determine the directionality of axonal growth cones. Combinatorial guidance cues permit exquisite spatial and temporal control of axon guidance, ensuring accurate wiring of neural circuits. The formation of synapses is a critical step in establishing functional neuronal connections. Synapse formation involves a series of intricate molecular events that ensure precise target recognition. Growth cones actively explore their surroundings, leading to the formation of transient synaptic contacts. Through a process of synaptic stabilization, specific synapses are retained, while others are eliminated. Cell adhesion molecules, such as neurexins and neuroligens, play crucial roles in mediating synaptic adhesion and specificity. Additionally, transsynaptic signaling molecules, including WNT and Notch, modulate synapse formation and plasticity. The functional connectivity of neuronal circuits relies on precise wiring and synapse formation. As axons navigate through brain regions, they encounter specific molecular cues that influence their growth and connectivity. Target-derived cues, such as neurotrophic factors, promote axonal survival and synapse formation, while activity-dependent processes sculpt functional connectivity. Activity-dependent mechanisms, including synaptic plasticity and long-term potentiation, refine and strengthen synaptic connections, enhancing the functional capability of neuronal circuits. The establishment of neuronal polarity is crucial for proper neuronal function. During development, neurons polarize into distinct axonal and dendritic compartments. This polarization is governed by various molecular mechanisms, including the localization of specific proteins and cytoskeletal remodeling. The axon initial segment plays a critical role in axonal polarization, acting as a gatekeeper for action potential initiation. Dynamic remodeling of the actin cytoskeleton and microtubules contributes to dendritic branching and neurite outgrowth, ensuring the formation of intricate neuronal networks. Communication between neurons relies on the precise release and reception of chemical messengers, known as neurotransmitters. These neurotransmitters are synthesized and packaged into synaptic vesicles, which are released by exocytosis upon depolarization of the presynaptic terminal. The neurotransmitters diffuse across the synaptic cleft and bind to specific receptors on the postsynaptic membrane, initiating a cascade of events that lead to signal transmission. Various neurotransmitter systems, such as cholinergic, glutamatergic, and GABAergic systems, play critical roles in neuronal communication and circuit function. Neurons exhibit remarkable electrical excitability, allowing them to generate and propagate electrical signals known as action potentials. Action potentials result from the opening and closing of ion channels across the neuronal membrane. Voltage-gated ion channels, such as sodium, Na+, potassium, K+, and calcium, Ca2+, channels, play a central role in action potential generation and propagation. The orchestrated activity of these channels ensures precise control of neuronal excitability and modulation of synaptic strength. The establishment of functional neuronal circuits requires precise sculpting of connections. During development, excessive synapses are formed, followed by a process of synaptic pruning, where weaker synapses are eliminated while stronger ones are retained. This activity-dependent pruning ensures the refinement of neuronal connections and functional circuit formation. Cellular processes, including apoptosis, engulfment of synapses by glial cells, and complement-mediated elimination, play critical roles in sculpting the developing brain. The wiring of the brain is a highly precise and complex process, dictating its functional capabilities. Molecular signaling pathways, neurotrophic factors, and activity-dependent mechanisms continuously shape and refine neuronal connections throughout life. The dynamic modulation of synapse strength and circuit connectivity allows for neuroplasticity, enabling learning, memory formation, and adaptation to environmental stimuli. The brain's wiring is a lifelong process, reflecting the intricate and adaptive nature of the human nervous system. Myelin, a specialized membrane produced by oligodendrocytes in the central nervous system, CNS, and Schwann cells in the peripheral nervous system, PNS, plays a crucial role in rapid and efficient signal transmission. Myelin acts as an insulating layer around axons, facilitating saltatory conduction and reducing energy consumption. Disorders affecting myelination, such as multiple sclerosis, can lead to impaired neuronal function and communication. Understanding the importance of myelin in healthy brain function is paramount in the diagnosis and treatment of these disorders. Decoding the complexity of the human brain is a grand challenge in neuroscience. Advances in imaging techniques, such as functional magnetic resonance imaging, fMRI, and electroencephalography, EEG, have allowed researchers to study brain activity and connectivity. Neuroinformatics, a multidisciplinary field, combines neuroscience, computer science, and statistics, enabling the analysis and interpretation of vast amounts of neuroimaging and genomic data. These approaches provide valuable insights into brain function, cognitive processes, and potential therapeutic targets for neurological disorders. In conclusion, we have discussed the intricate processes involved in neuronal development, including cell navigation, connectivity, and signaling. This knowledge is essential for understanding the structure and function of the nervous system, enabling us to diagnose and treat neurological disorders effectively in our medical practice.